Nice.
Andre's handling the Facebook there.
Yeah, the Facebook's streaming now. So.
All right, let's go ahead and admit these real quick. Hello, everyone. Welcome. We're so glad you were able to join us for today's webinar. It's our pleasure today to introduce Dr. Kailinda Bauer, a J.N. Andrews Honors Scholar. Dr. Bauer graduated from Andrews University with a BA in Music and Bachelor of Science in Biology in 2014. She recently received a PhD in Microbiology and Immunology from the University of British Columbia, Canada. A veneer recipient, Kailinda's doctoral research in Dr. B. Brett Finley's lab explored how gut microbes influence early life malnutrition. Additional research experiences include projects at Harvard University, Lee Kong-Chien School of Medicine in Singapore, and Lleida Universidad in Spain. So we'll go ahead and turn the time over to Dr. Bauer. Welcome.
Perfect. Thank you so much for joining. I'm just going to go over and start screen sharing now. I'm sure Laura or Andre will tell me if the slides can't be seen, but hopefully you're seeing what I'm seeing, an intro slide. So again, thank you so much for joining in. I'm so excited that I can join the alumni homecoming events virtually, and I'll be discussing my research looking at how gut microbes might impact health. And as was mentioned, I am rather a recent graduate from the University of British Columbia, and I am also part of the alumni family at Andrews University. I graduated in 2014, doing degrees in biology and also music. So I've always really loved research. I've loved investigating health and how microorganisms play a role in health and diseased states. And as a J.N. Andrews Honors Scholar, I had the chance to do a senior thesis where I was looking specifically at gut microbes and how antibiotics impact their function. And as was mentioned before, I've also had the opportunity to do research kind of globally, in addition to some of the places that Laura mentioned. I also have worked with USAMRIID, so the US government, and also studied and done research at Walla Walla University's Rosario Beach Marine Laboratories. Well, today I'll be kind of discussing the gut microbiota, who's there, what are they doing, why are they important? And to kind of answer that last question, I'll tell two stories from my own research, both looking at the gut-brain axis and also the gut-liver axis within the context of a disease state, undernutrition. And then I'll have some concluding remarks, and of course, any questions, feel free to ask them at the end or send them in the chat. I'm not sure quite how this is working. So in 2007, the National Institutes of Health, NIH, funded the Human Microbiome Project. And this was a collective, collaborative effort to characterize microbial communities that reside throughout our bodies. In 2012, they published a landmark paper in the journal Nature, where they gave some of their earliest findings. Scientific American that same year created this interactive graphic to look at gut microbes and at the human body. And from what hopefully you can see in this graph, that we are colonized by many different microbes throughout our surfaces, both the external surfaces like the skin, and also internal surfaces if you think about the respiratory system, urogenital tract, and also the gut microbiota. So when I'm using the term gut microbiota, I'm referring to the collective microbial community that resides along the gastrointestinal tract. And also, hopefully from what you can see from this image, that this microbial community is both the largest and also the most diverse community that we have in our bodies. Perhaps you've also heard the term microbiome. So microbiota and microbiome differ Slightly in precisely what they mean. Microbiota is referring to the organisms themselves, and microbiome is referring to their genomic content. So think about their DNA that they're bringing on to the human system. For example, if I was to pick a department at Andrews University, I could list all of the different professors, and that might be the microbiota. But if I say that this professor is a chemistry professor, this is a biology professor, this is a physics professor, and this is how they're influencing the STEM program, that could be kind of correlating to the biome aspect, what they bring to the table in terms of teaching. Another term I'll use in this talk is the metaorganism, and this is a concept of individuality. So instead of thinking about the human as a single individual, I'll be referring to them as a metaorganism. So not just the human component, but also the collective microbial communities within the human. And I'll touch a bit more about the metaorganism towards the end of the talk. And so when we look at the gut microbiota, there are many different microbial residents. You are colonized by viruses, by yeasts, by single-celled organisms called archaea, and also by bacteria. And out of these different types, bacteria are the best characterized and most studied. And gut bacteria extend human function. They extend our digestive function. They can break down complex starches and carbohydrates in our diet. Microbes can also influence our immune system development. They also synthesize essential vitamins, such as vitamin K and vitamin B. Certain microbes can also metabolize specific drugs that we take in our body. They are also influential in our metabolic functions and in our fat storage processes. And then here you're seeing two neurons. This is a synapse, so a neuron-neuron connection, and also a behavioral test with a mouse in it. So the gut microbiome also informs our brain and behavior. And I'll be discussing two stories, mainly looking at these functions. So if you were to peer into a healthy gut, you would hopefully see heavily colonized epithelial and mucosal spaces with a highly diverse group of microorganisms. But when researchers examine disease states or the dysbiotic, altered, impaired gut, this is most often characterized by a loss in diversity and sometimes a potential bloom of pathogenic microbes. So for example, the most studied or one of the most studied disease states with the gut microbiota are the enteric diseases. Here I'm showing a study of inflammatory bowel disease. You can see this final column here is showing the healthy states, and the first four are looking at inflammatory bowel conditions, ulcerative colitis, and also Crohn's disease. And you can see that for certain microbes, they are decreased in inflammatory bowel conditions, but they are increased in the healthy states. Now, obviously, this data is showing a correlation. That is to say, you have a disease state, you have a change in the microbes, and then you have a healthy state and you have a different change in microbes. So we're not saying that alterations in the microbes are causing the disease. We just see a correlation between certain decreases and increases between healthy and diseased conditions. So are alterations of the microbes informing pathogenesis of disease? Or do you have an inflammatory bowel condition where you have inflammation, you have an altered immune state in the gut, you might even have damage of the different villi within the intestinal tract, you might be on medications. Are these changes altering the gut microbial communities? Well, it's probably a combination of both, but we do have some evidence that gut microbes themselves are important for disease progression. For example, you can take the case of neurodegenerative diseases. The Finley lab where I did my doctoral research also studies Parkinson's disease. So Parkinson's disease is characterized by slowness of movement, by rigidity, by tremors. You have a loss of dopaminergic neurons. But even before a patient might receive the diagnosis of Parkinson's disease, you start seeing symptoms that are associated with a later progression of Parkinson's disease much earlier. So even decades earlier, these patients often display signs of constipation, so an altered gut state. So we can also do experiments to kind of look and pinpoint potential causative impacts of gut microbes. Some of the most well known of these projects are these germ-free studies. So Louis Pasteur, who is a French chemist, a microbiologist, one of the fathers of bacteriology, once wrote that life would not long remain possible in the absence of microbes. And I'll take a little aside detour to discuss about Pasteur's education and his early start in microbiology. So when he went off for his secondary education at the Collège de Besançon, he actually failed his baccalaureate in science, and he had to repeat a year of mathematics. So if you are listening to this and you are struggling with your STEM classes, or if you teach someone who is struggling with STEM classes, or if you have a child or grandchild who is struggling with math or science or biology, just know that one of the greatest microbiologists, Louis Pasteur, failed, and he went on to do great things. So stick with it. So he writes this famous phrase where, according to Pasteur, he believed that if we have microbes in our body, then they must be important to us. And so if we didn't have them, he hypothesized that we could no longer live and function. Well, even in this statement, he was incorrect, because scientists and researchers have shown that you can exist without a microbial community. For example, in rodent studies, you'll have mice, and just like any other mammals on Earth, they will be colonized by bacteria following birth. But then you can also have germ-free animals, and these are mice that are the bubble animals. They are sterile, they are, in a sterile environment. They receive irradiated food, so they have no microbiome. And their offspring will also have no microbiome as long as they are kept within a sterile containment. So researchers will do experiments where they will compare mice who are raised conventionally, who have a gut microbiome, versus mice raised in germ-free conditions. And so if they see a disease state or any alterations, what they can do is then introduce microbes into the ex germ-free mice, and then see, do we see a change in the conditions, or do they remain the same? And if there's a change, this can indicate that gut microbes are important for this particular health function. One of the most famous studies involving germ-free mice was work done out of Jeff Gordon's lab by Dr. Vanessa Radara. So in this study, the Gordon lab examined a twin cohort. So they looked at many different twins, where one twin was obese and one twin was lean. And from the obese twin, they took fecal stool samples, and they took the microbes from these and placed them into germ-free mice. And then they also took fecal stool samples from lean twins and again placed them into germ-free mice. And what they saw was, after mice received an obese microbiome, these mice ended up gaining more weight compared to mice that had received the lean twins microbiome. So even when the mice were kept on the same diet, having the bacteria from an obese human increased their adiposity, or their fat deposits within the mice. And so this also seems to imply that gut microbes are important for our metabolic processes. Now, mice are coprophagic, which means they eat poop. So if you have mice that are in the same cage, if one mouse poops, another mouse might come over and start nibbling on these fecal pellets. So they were also interested in using this characteristic of mice to examine transmission of the bacteria. So they co-housed mice that had received the microbiome from the obese twin, and also mice that had received a microbiome from a lean twin. And they found that when the mice were together, the obese microbiome wasn't really able to effectually pass over into the lean microbiome mice. However, you had these lean microbiome mice, and after a period of co-housing, mice that had the obese microbiome also had bacterial signatures of the lean microbiome. So it seems like health, in a way, was able to transfer in this condition. And we think about metabolic conditions like diabetes and obesity as non-communicable diseases. So this is in contrast to infectious diseases where, if you have a cold, this is something you can pass on to another person. But if you say, oh, I have this one condition, I really can't pass it on to another person. Now, perhaps the microbiome is opening avenues where we can look at diseases that we classify as non-communicable and say, well, hey, can we alter bacterial states? Can we alter transmission patterns? And maybe we can see a different outcome in health. Now, of course, these are animal studies. They are done in highly unrealistic conditions where you have exact foods, you have exact housing conditions, you have exact bacterial transfers. So there still needs to be a lot of translational research ahead to see whether these findings can be applicable to humans. So hopefully from this talk so far, I've emphasized that the gut microbiome is a very diverse, highly complex, highly dynamic feature within humans. So how do bacteria that are residing in the gut have these systemic impacts? For example, in Gordon's group, how are they influencing fat deposits and also liver function? Well, we talk about major pathways of how bacteria influence these peripheral organs. Here I'm showing just two graphs of the gut-brain axis and also the gut-liver axis. And I just want to emphasize the dynamic complexity and how there are many different mechanisms and pathways involved in these interactions, from metabolic pathways to immune pathways to inflammatory pathways to even neurological pathways. So my work, my doctoral studies focused on kind of the other spectrum from research in the Gordon's lab, because I was looking at undernutrition. So we know that malnutrition is the leading contributor of deaths in children under the age of 5. In fact, recent estimates state that as much as 45% of childhood mortality can be attributable to malnutrition. And although malnutrition rates are going down, with COVID-19, the pandemic, and also the refugee crisis, we're starting to see spikes in early life undernutrition. And beyond mortality, beyond growth deficits, early life chronic undernutrition results in these long-term health consequences from altered microbial states, altered digestive processes, poor metabolic function, immune disruptions, and also an increased risk of having neurocognitive deficits and brain impairments. Now, undernutrition and these lasting consequences of undernutrition don't solely exist within the world of nutrition. You have all of these external factors that are playing a role. For example, let's take cognitive impairment. You have a dietary component that is influencing cognitive capacity, but you also have socioeconomic status, you have access to education, political stability, even maternal and paternal education levels. All of these will play a role in academic performance of children who are undernourished. One of the other great biological factors that plays a role with how undernutrition impacts long-term health is an umbrella term that we refer to as fecal-oral contamination. So this occurs in regions where you have poor access to sanitary water, where you have increased exposure to enteric, that is gastrointestinal infections and diarrhea. You have more exposure to fecal microorganisms. You also have poor access to proper sanitation services. In fact, around 2 billion people around the world only have access to pit latrines. So these are children who are undernourished, but are also in frequent exposure to fecal microbes. So what does fecal-oral contamination do? Well, if you have an infection or inflammation in the gut as a result of fecal-oral contamination, you have damage alongside your gastrointestinal tract. And this results in a decreased nutrient absorption. So of course, this exacerbates the consequences of malnutrition, which in turn increase susceptibility for these microbial alterations. So people that are living within this condition are trapped within this perpetual cycle of poor health outcomes. And these have been linked to cognitive impairment later in life. Well, the Finley lab developed a mouse model that combined both this concept of fecal-oral contamination as well as nutrient deficits. This was known as the MalBG model, or also you'll hear later on in the talk, MBG, it's the same thing. So in this model, you have a combination of a malnourished diet, which was a protein and a fat deficient diet, also with bacterial fecal exposures. And then we're comparing this against a group of mice that are kept on the malnourished diet without the gut dysbiosis from fecal-oral contamination. And then I also have a healthy control, which are mice that are maintained on a standard diet since weaning. So we had this model and we were interested to see whether we could demonstrate or show that both diet as well as gut microbial alterations combined could play a role in cognitive impairment later on in life. To do this, we focused on a specific cell known as microglia. So within the brain, within the CNS and the brainstem, you have neurons. These are the famous cells of the brain, but you also have these assisting cells, the glia cells, and microglia are a subset of glia cells. They look like this. Here I show an image in the center where you have your cell body, and then you have these long dendritic processes, these arms that extend out from the cell body and are constantly moving to survey the environment. Now, microglia have many different functions. They have an immune function where they use their processes to scavenge debris. So if they see cellular debris, if they see a potential pathogen that has crossed over the blood-brain barrier, if there is any type of oxidative stress going on in the brain, these processes will move towards the damage, they'll cordon off the damage, and sometimes you can even see this formation of these round circular lasso-like structures, which state that the microglia are phagocytosing or chomping or eating up the pathogenic insult. But they also use this eat me quality early in life where they're involved in brain plasticity. So when you're born, you have all of these neurons that are connected to each other. So you have these many different neuron-neuron connections. But as we develop, certain neuron connections that we no longer need get snipped away, and microglia are involved in these removal processes of certain synapse connections. So this is very important for brain plasticity, or how well your brain can adapt, learn, relearn, forget different stimuli, memories, or functions. But also what was important was that the year I started my PhD, Nature Neuroscience published a paper, again doing a germ-free study, which demonstrated that gut microbes were very important for how microglia develop and how they function. What's also really exciting about microglia is that they have very unique morphology depending on what they're doing in a specific situation. So again, as I mentioned, in a healthy state, microglia will be responding to different signals from the brain, and they have these long processes. You might see some of these eat me lasso-like phagocytic structures in the microglia. However, under chronic stress, when you have all of these potentially inflammatory or oxidative or pathogenic influences impacting the microglia, these processes will retract and they'll have more of this amoeboid-like form. And so my research was looking at how microglia responded to results. Here you're seeing microglia respond to a lesion or a damage within the brain. And then also looking at some of these phagocytic properties and how they're influencing health and function. And so this research is currently under review. So just a snippet, a preview of hopefully research you'll be able to learn more about in the upcoming future. But another interest for my research and also the Finley lab was how gut microbes and how malnutrition are involved in our metabolic functions. And I was specifically looking at a condition known as NAFLD, or non-alcoholic fatty liver disease. So here I have this graphic, and on the top, you can kind of see macroscopic, I guess, images of the liver and healthy liver. It's nice, bright and red. Underneath, I'm showing an illustration of hepatocytes. So hepatic referring to liver. These are the cells, the major cells of the liver. And you'll see in a healthy condition that the cells are nice and tight and packed together. But in the absence of alcoholic insult, when you have increased fat deposits in the liver will turn more of a dun color, so more pale colored. And you'll also see this fatty buildup within hepatocytes, so they'll balloon up. Actually, a quarter of the global population presents with NAFLD, and it's a subclinical condition, so you might not know exactly that you have NAFLD or that it's influencing your life. But what's scary about NAFLD is that a quarter of these cases can progress onward to more damaging conditions, including non-alcoholic steatohepatitis, where not only you have the fatty buildup, but you start seeing cell death and also inflammation in the liver, and even up to cirrhosis, which is an irreversible liver damage, which also now has fibrotic scarring on top of these earlier insults. However, most of the research into NAFLD examines this disease from the perspective of overnutrition. So NAFLD is tightly linked with metabolic syndrome, with obesity, diabetes, weight gain, aging. However, we also see NAFLD in cases of undernutrition, and perhaps the most well-studied case of undernutrition and NAFLD is kwashiorkor, and this is a specific undernutrition form, which is often diagnosed shortly after children have stopped breastfeeding and are moved on to a solid diet. You see wasting, that is like low muscle mass and integrity in the extremities. And then you also see the characteristic edema or fluid retention within the abdominal section. So they have a bloated belly appearance, but also an undernourished appearance as well. And one of the features of kwashiorkor is this evidence of a fatty liver. So our mouse model also shows signs of fatty liver features. Here you're seeing a liver from a healthy mouse, and here I'm showing microscopy of these hepatocytes. You can see they're nice and tightly packed together. And here's our malnourished condition in our MalBG or MBG model. And you can see some of these cells here have this balloon-like appearance where they have these fat deposits within them. And also when I measure liver triglycerides, or these small fatty acids that are important for how we store fat and how fat gets passed throughout our body, I see that these are increased in the malnourished states and also in the MBG state. One of the big questions in our lab, and for me personally when starting this research, was we have a condition where we can now characterize specific features of undernutrition, where we can look at the role of diet and also the fecal microbiome. But what we're most interested in and what we're most excited about are, can we actually reverse features of undernutrition? To do this, I looked at a dietary reversal study. I'll show some of these results right now. On the top, I'll have my healthy control mice. These are ones that are maintained on that healthy diet. And then we have our MBG model, again, retained on a malnourished diet. This bottom figure here, I'm showing a group of mice that I called MBGR. This is our reversal of malnutrition. So they start off on a malnourished diet, and then they're placed on a healthy diet. And then I also have a group of mice that I'm adding called the CMBG mice. And this is an adult onset malnutrition. So these mice are maintained on a healthy diet early in life in a pediatric state, but then in the adulthood state are placed on the MBG model. So we were very excited to see that dietary interventions alone improved growth faltering. So I'm showing average weight of the groups across the weeks of the experiments. And you can see all mice start off with the same weights. And as they move on, the control or the healthy mice in these dark black line gain significant weight compared to the undernourished mice. But later on in this reversal phase, you can see that when mice are placed back onto a healthy diet, this dark purple group here, they start gaining weight. So by the end of the experiment, even though their average doesn't quite reach that of mice that have been healthy long-term, it is no longer significantly different from each other by statistical measures. But what about conditions of undernutrition like NAFLD? So here again, I'm showing histology from our healthy and our malnourished mice. So again, you can see the healthy state, the nice tightly packed hepatocytes, the liver cells. But in the MBG state, you can see hepatic steatosis, which is again, the ballooning, the fat accumulation within the hepatocytes. What was very exciting was in our MBGR state, the hepatocytes now look very similar to the control or healthy mice. And what was intriguing for us was in the adult onset malnutrition, again, we see histology that more closely resembles the healthy mice than the malnourished mice. And for us, this seemed to signify that early life diets are very important. And so if you alter or you change something in early life, you might have these long-term consequences. But if you have these insults later in life, in adulthood, you might have a more protective outcome where you don't quite see the exacerbated consequences from youth and pediatric conditions. Again, we measured fat accumulation within the livers, specifically the triglycerides. And even though the histology of these CMBG mice looked close to the healthy mice, we can see that diet really plays a role in the fat accumulation within the liver. However, dietary intervention reduced non-alcoholic fatty liver features in our malnourished mice. But we were also looking again at the fecal microbiota community. There had been a paper published a couple years ago in Nature Medicine that identified specific bacterial pathways that were linked to NAFLD in cases of obesity and overnutrition. So we decided to look at some of these bacterial pathways in our undernourished mice. So if the pathway was high, it was linked to NAFLD, and if it was low, it was linked to a healthy condition. So when I looked at those same pathways in our mouse model, or the predictive pathways of what we're saying these gut microbes are doing, we saw that they were increased in the MBG state, but placing on the dietary intervention reduced the frequency of these pathways in the bacteria, which again seems to suggest that perhaps the gut microbes themselves might be a therapeutic target where we can alter microbial communities, and in doing so, maybe have better outcomes for NAFLD cases in early life undernutrition. So some takeaways from this talk. Hopefully you found some interesting things in the doctoral research. But if not, hopefully I've managed to convince you that we should be valuing interdependency, recognizing that we exist in this microbial world, that microbes can be beneficial for us, and that in fact, they are necessary for certain functions for this optimal health condition. So just as we value interdependency and diversity in academic, in social and community situations, again, just even within our own bodies, our microbes play an important role, and we house diversity within us. Microbes are not only involved within in the gastrointestinal tract and impacting our digestive and enteric functions, but they also have these systemic influences. However, the precise mechanisms of how a certain bacteria or bacterial product influences peripheral organs is still largely unknown, and we're very much in the infancy of looking at microbiota studies and determining the precise consequences of bacteria or bacterial communities. And then with my doctoral research, that undernutrition and this condition known as fecal-oral contamination, where you have overexposure of fecal microorganisms, they seem to play a role in the outcomes of undernutrition, looking at both liver and brain function. So I also want to emphasize this caution regarding microbiome research. If you just turn on your television, microbiome has become such a hot topic. You can see soaps or shampoos that are microbiome friendly, or certain foods that are promoted as being beneficial or helpful for your microbiome. In fact, if you go into certain drugstores, you might see a collection of medicines that say, oh, look, we can alter your microbiome, we can help cure depression, anxiety. And I just want to say there's a big caution around this, because we still don't know what is a beneficial bacteria. I'll give one quick example, and that is a microbe known as Akkermansia muciniphila. So Akkermansia muciniphila had this inverse relationship with obesity and metabolic conditions. So once you had, so this bug was associated with, okay, if there's low levels of it, then you might have an increased risk of all these metabolic conditions. So people thought, oh, well, all we need to do is add this bug back in and we can get healthier outcomes. And they showed this even in mouse studies. Adding these microbes in helped protect the mice from gaining weight and having metabolic features. So let's go ahead and let's package this bacteria up. We can send it to people. It could be beneficial for these conditions. However, our lab and many other labs have also shown that if this bug is in high levels, there's an increased risk or correlation association with Parkinson's disease, which no one wants to increase your risk of neurodegenerative conditions. So in one condition, this bacteria was seen as a good bug. In a different condition, this bacteria was seen as a bad bug. So we're really still not quite sure how bacteria and how they're interacting with other bacteria and in the gastrointestinal milieu might be impacting health consequences. So don't go and buy a certain bacterial cocktail thinking that it will cure or reverse specific conditions. However, there are ways that you can support a healthy microbiome. And many of these involve proper lifestyle patterns that we promote at Andrews University. So first, eating a diverse and healthy nutritional sources. I do want to mention that when you hear the terms prebiotics and probiotics with foods, prebiotics refers to foods that are beneficial for microbes. They are microbial foods. Examples of these can be legumes. Also, honey is a prebiotic, cashews, walnuts, certain bean products. Probiotics refers to a food that has bacteria within it. So this could be fermented food products like kimchi or yogurts. So what's a bit tricky about probiotics is that even if you were to, let's say, digest or take in yogurt that has all of these different bacteria, we still don't know how much of this bacteria makes it through the stomach acidity and actually colonizes our gut. So the debate about probiotics remains. However, there have been studies that show that having more prebiotics and probiotics can have, in certain situations, a beneficial outcome. But mainly you want to be trying to eat a very healthy, diverse food group. So have lots of plant-based products, have a variety and a different variety of vegetables and fruits within your diet, not just maintaining, oh, I'm only going to eat this one food item and keep and keep eating it throughout the upcoming weeks. So try new things. Exercise and maintaining a healthy sleep schedule has also been linked with having a diverse, which we often term as a healthier microbiome. And then explore. Unfortunately, because it's the year 2020, we can't really maintain these community interactions that we have with each other that are linked to beneficial transmissions of microbes, but we can still introduce our body to microorganisms by exploring nature and spending time outdoors, by maintaining social distancing, but also interacting with our environment, especially the natural environment, in appropriate and also healthy ways. So my research, my doctoral studies were funded by the Vanier, also by CFAR, the Bill and Melinda Gates Foundation, also supported our lab's research. And I'm very thankful for researchers in the Finley lab, MacVicar lab, and Ayala labs, both at UBC and also at Lleida University in Spain, for supporting my research. If you want to learn more, I will shamelessly self-promote myself. I gave a TED Talk a couple of years ago that you can find online. I also have a blog called The Scope, where I publish graduate life and also research that I find interesting in the microbial world. If you're more interested in kind of an academic take on how we have a gut systems perspective on health, I have a review that was published in Bioessays last year, looking at the gut-brain axis, and also some research that was recently published, which tells more about the NAFLD and undernutrition story. So thank you very much for joining. I'm so glad I could talk and say hello to everyone from afar. That wraps up my talk. I'm open for any questions.
Thank you so much, Dr. Bauer. That was fascinating. I have to say, I learned a lot. I think everyone viewing did as well. So let's go ahead and move into the question and answer time. Just a reminder for those of you here on the Zoom chat, go ahead and type your questions into chat. And for those of you viewing on Facebook Live, you can also comment or message us, and we'll monitor that and pass questions along to Dr. Bauer here. First question we have is, what is your perspective on probiotic supplements?
I'm always a bit hesitant to answer health-related questions because I do not have medical training. I can discuss some research that has come out. There has been studies out of the Weizmann Institute and the Elanov lab that were looking at how probiotics benefited or harmed patients after having a load of antibiotics. So sometimes we need antibiotics, whether we're going into a surgery and we want to prevent infections, whether we have an infection and we need a bacterial infection, not a viral one, but a bacterial infection, and we need an antibiotic course to remove the pathogens. So once we do that, unfortunately, many antibiotics also harm other potentially beneficial microbes in our gastrointestinal tract. So researchers thought, well, why don't we give antibiotics, but at the same time be providing probiotics, so we're adding microbes back in? And the Elanov lab said, well, wait, does this work? Like, can you take a probiotic supplement and all of a sudden gain diversity in their microbiome? And their research actually showed the opposite. If you didn't take a probiotic, you jump back much faster to a healthier state. However, we also have lots of research that has been done looking at specific lactobacillus or bifidobacteria. These are bacteria that you would commonly find in yogurts or probiotic supplements. And in mouse studies, these were very beneficial in reducing inflammatory conditions and even impacting certain brain features. So for probiotics, if this is something that you have a specific yogurt brand that you really like to eat in the morning, I am a big fan of yogurt. I'm a big fan of frozen yogurt, so I partake of yogurt quite frequently. If you are taking the yogurt and you feel healthy and you feel fine, then I say go ahead. But I think in the upcoming decades, there's going to be a lot of research in trying to figure out, can we actually create a bacterial community that we can guarantee is beneficial and will colonize the gut? And I don't think we know that yet.
All right. Next question is, on your research, why did you differentiate between MBG and CMBG mice, the last slide before your conclusions? Can you speak to the statistically significant difference in your data?
Sure. I'll just go back. I can even just show this. Maybe this graph will be a little bit easier to see. So the MBG mice are mice that are placed in our malnourished model right at weaning. So all of these mice will start their prospective diets right after they have moved away from mother and are starting on solid foods. So MBG are maintained both in a pediatric condition and an adult condition on a malnourished diet. The CMBG mice after weaning are placed in a healthy diet. So they have their childhood, their pediatric space in a healthy diet. And it only isn't until they've passed sexual maturity that they are placed on an undernourished diet. So what I can do is I can run a statistical test like an ANOVA, which looks at variance between the different groups to see if there's a difference between a healthy state and the different undernourished states. So hopefully that answered the question. I'm also happy to chat more later.
Sounds good. Next question is, what are you interested in researching next?
Well, my upcoming postdoctoral research will be looking at liver cancer and the gut microbiome. So I'll be interested in how bacteria are altered by cancer conditions and how altering bacteria might benefit cancer outcomes. So I'll be keeping one foot in microbiome research, but taking also a big step in a new field.
Sounds like it'll be an interesting area to learn more of in the future. Next question is, do you have any data relevant to the effect of FOC on the behavior of your mice? And the commenter said, had a comment that he thought the mice would be less vigorous and active than mice lacking such fecal-oral contamination.
Can you repeat that?
Sure. Do you have any data relevant to the effect of FOC on the behavior of your mice?
Yes. Short answer is yes. There are both behavioral differences as well as cognitive differences. I can say that fecal-oral contamination seems to exacerbate neurocognitive impairment. And hopefully, if I'm invited back in a couple of years, I can present that data as well.
Next question is, how far are we from providing the general public with a genomic analysis of the biome analogous to the popular human genome analysis, such as an analysis that would allow comparison of individuals' biomes and the diseases associated with them?
Well, the answer is both. It's already here and it's upcoming. So if you were to type in the Human Microbiome Project just online, you can go to the National Institutes of Health, and all of the data of different microbial communities within healthy subjects has been published and is online. So you can look at hopefully the healthy microbiome of the skin, of the mouth, of the lungs, of the gastrointestinal tract, etc. What is a little bit tricky about microbiomes compared to the human genome is that when we are born, we have, our DNA, and that really doesn't change. I mean, it can change. You can have mutations. You can, have cancer. But the microbiome compared to our DNA is so highly dynamic. If you travel to a different country and eat different food, and you compare a fecal stool sample from post-travel to pre-travel, you will see differences. So we're still figuring out whether there is a core microbiome, and there have been publications that have looked at tens of thousands of humans and have identified bacteria or bacterial signatures that they say are these core signatures for health. But there's also this recognition that it is really highly dynamic, that depends on your environment, on your interactions, on whether you're sick, on whether you start taking medications, whether you go off medications. All of these can influence the bacterial communities within your gut. So we're now shifting from who's there to what are they doing. So what are their metabolic pathways? And these can be more stable than the bacteria themselves. So for example, if you have someone who, let's say, is an artist, and someone who is a sculptor, and someone who is a musician, they are all doing different artistic tasks. If you remove one from the community, you might lose that feature. So you might have music and paintings, but no sculptures. But you could also remove one from the group, and a bacteria could be there, or in this case, an artist could be there who says, oh, I actually also like making sculptures as well. And they suddenly start producing sculpture and art and music. And so you don't see this loss of functionality. So we're starting to, or we have been really for the past decade, looking more at the functional changes versus who's there and who's missing.
Next question is, what implications do you think your research could have in the future on how we, both as a society and as individuals, provide humanitarian aid, considering the malnutrition aspects?
One thing that surprised me when I was looking at undernutrition studies and interventions, we have, or one thing that's often given to severely undernourished children are RUTFs, so ready to use therapeutic foods. So an example could be Plumpy Nut. So there's often these like peanut pastes or high calorie, high rich foods, often in a paste-like material, that are fed to early life children. But what surprised me when I was looking at some studies was how short-term these dietary interventions were. So a child would come into a clinic severely undernourished. They'd be given medications to kind of improve infectious conditions. So let's say, you have a bacterial infection, they're placed on antibiotics. They're given this dietary intervention for a week to a month, and then afterwards sent back to their environment. So it's really tricky because, if you go back to your home environment, which is in a condition where you are surrounded by malnutrition or fecal-oral contamination, you might not have access to appropriate nutritional sources. So what's exciting about microbiome work and showing that we can alter the microbiome to influence or promote health means that even if we couldn't maintain a pediatric population on the ideal nutritional context, if we can have a microbial focused therapeutic, this might also result in better health outcomes. And also there has been a lot of research by the Bill and Melinda Gates Foundation to look at ways where we can reduce fecal-oral contamination. So there are projects known as the WASH trial and also the SHINE trial, which not only gives nutritional interventions, but also tries to remove fecal-oral contamination by providing better access to latrines, to coming up with innovative ways to put low cost toilets into communities, with also promotional and educational material on hand washing and food preparation. So I think, with mice, it's on one hand, I can do all of these factors and change these experimental conditions to get a result that's favorable within a clinical, within a pediatric population, there are so many external forces that play a role in our health and our health outcomes. If we can make some even minor tweaks that promote better outcomes or decrease mortality, I think that will be a really exciting outcome for my work.
I think we have time for one final question here, and then we should probably wrap this up. The question is, in relation to medical trials that try to control for gender, age, comorbidities, etc., given the role of microbiota on drug performance, do you think it's time to start controlling for microbiome difference? And a follow-up with that was, how much would you take a wild guess on we might not be controlling?
I would think that would be almost impossible to control for microbes. I mean, if you were to look at all of the thousands of different microbes that could be different between people, and even within a bacterial member, there are different types of strains that can be classified as one species of bacteria. So you might say, oh, I have an E. coli, and this is a commensal in me, and another person can say, oh, I have E. coli, and it's causing me disease. So it's just so dynamic that I think if you were to control for every single bacterial alteration, you would easily lose significance in clinical trials. Perhaps if there was a specific bacteria that you were interested in, you could control for the relative abundance of that one bacteria or this collection of bacteria in your trials. But for a full gut microbiota exploration, I think that would be very difficult.
Great. All right, thank you so much for your time. Now, if we have more questions, can you go back to the last slide on how participants can learn more? And
Absolutely.
do you have a suggestion on how we might be able to send you a couple of follow-up questions that we still have unasked?
Sure. If it comes in through Andrew's alumni, you can send it over through my email. I'd be happy to answer more. I think also on my blog, there's some contact information there.
Sounds great. Well, thank you very much for your time, Dr. Bauer. This was, like I said, fascinating, and I can tell that everybody, I think, really enjoyed hearing your research and your findings with this. For everybody watching, we have one more event coming up this afternoon that will be at 2:30. It's a virtual healthy cooking demonstration, so that might actually go right in line with the healthy theme here that Dr. Bauer has spoken on. So we hope that you'll join us for that. It's going to be streaming on alumni.andrews.edu/celebrateau, our landing page there. And we'll have a live Q&A for presenters afterwards as well. Thank you again for your time, and hope everybody enjoys the rest of your afternoon here.